So when working in the lab, you must wear full PPE, and that must be a lab coat and latex gloves. Each lab practical will have a risk assessment and COSH paperwork, which includes the chemicals in use. All this paperwork is found online, and if you ever have any doubts, the lab practicals are supervised by members of lecturing staff, and you must ask questions if you ever have any doubts or any questions you may need answering. Today's tutorial is to look for saliva onto exhibits such as balaclavas, items of clothing. The test we use is the Faddy-Bass test. This is known as Faddy-Bass paper. On one side there is a blue starch granules, and on the other side is just plain white paper. In saliva there is an enzyme called alpha amylase which is present in high volumes in saliva. The chemistry and the biology involved in this test is the amylase starts to kind of break down the starch granules, and in doing this releases this blue dye which then helps you to localise an area on an exhibit. Saliva you can't see, so we need to have a test, a presumptive test, to help us to find where the saliva is on an exhibit. The first step of the procedure is to test if the Faddy-Bass paper is working optimally. And how we do that is we use control samples of the Faddy-Bass paper. And first we apply a known saliva sample, and we apply this, we use 30 microlitres of a known saliva sample onto a fabric, a pre-prepared fabric. And we dispense the tip into our biohazard container. We also have another piece of fabric, and we want to just put water on the fabric, and this is known as a negative control, to see if it just reacts with water. And we apply the same amount, which is 30 microlitres, on the fabric and dispense the tip safely into the biohazard container. We then get our Faddy-Bass paper, and on one side it has the starch granules and it's white on one side, and we want to put it the blue side down, the starch granules down, and we do the same for our negative control. So now the paper is making contact with our fabric. We just get a piece of acetate, just clear acetate plastic, and we apply this over our samples. And then we get quite a heavy weight, about 3 kilograms, and we place this over our sample, and we leave that to kind of react and for up to 5 minutes, and we'll see the reaction to see if the positive control has worked and if the negative control has not reacted, so then we can have confidence in the Faddy-Bass paper that it's working as it should be. So once the 5 minutes is up, you want to take the weight off, put it to one side, and then you want to review your controls. So the positive control, as you can see, this blue dye here has kind of leached through the paper, which is a positive. Yes, it has broken down the starch, so that's great. And on the negative control, we do not have any blue colour here. So the positive and controls and the negative control has passed. So now we can proceed to actually examine the exhibit. So we have our exhibit here, and the first thing to do is we look at the exhibit to make sure that there's no holes and the packaging's integrity is there and it's been sealed properly. We then use our examination notes and we look at all the information the CSI has put on the label, and we use our documentation here to write down everything which is on there. Once that is done, we are then happy to enter the bag. So we always open the bag, we never open on somebody else's seal. So we just basically use a pair of scissors, cut to the side, and then we get our item out. In this case, it's a piece of clothing, and we place it on our examination bench. And in this case, it's a balaclava type kind of clothing. And we do our documentation of describing the item to see what condition this is, the condition of the item, if it's new, old or worn. Once we're ready to carry out the actual saliva Fadibas test, we have a look with our eyes. We use torches to see if we can see any kind of obvious kind of white staining. In this case, we can't see any white staining, so we are going to use the Fadibas test. So the first thing we do is, because it's kind of 2 layers of fabric, we place an acetate in between the balaclava, like so. There we go. And now we are ready to actually place our piece of Fadibas paper. So you can cut this to different sizes. So if it's a larger item, you'd have a larger piece, etc. And as you can see, we have the starch granules on this side. We then place it onto our item of interest. In this case, you've got 2 eyepieces and a mouthpiece. So we're going to cover up, obviously, the area of the mouthpiece. We use water, so just normal sterilised water, and we spray this onto the Fadibas paper. Remember to put the Fadibas paper blue side down, the blue side making in contact with your item. And you don't want to saturate the paper, but you kind of want to put water on there so it covers the whole surface area. This might take you 10 seconds or so to actually apply the water. Once you've kind of covered the total surface area, you then get a piece of just plastic, some acetate, some clear acetate paper, and you place this onto your Fadibas paper. You kind of nice firm kind of contact, and then like you did with the positive control and negative, you place your weight over your item, and the total time set is 40 minutes. So the total reaction is 40 minutes in length, and in 10-minute intervals you should check on your Fadibas paper and update your examination notes appropriately to see if it's reacted, if it's changed coloured. So the full test is 40 minutes, because anything after 40 minutes is classed as a false negative. It's basically a false result. So we have to wait for the full 40 minutes time. So once the 40-minute time is actually up, you remove the actual weight. Be careful, very heavy, place to one side, and then you want to look at your results. So on this here, it's clearly showing a blue diffuse stain, like we saw in the positive control. And if you peel back the actual acetate, you can actually see this diffuse stain. What you have to do now, using a clean waxed crayon, is carefully, you can pull this back, and very carefully, using your positive area, is outline where there is alpha amylase. So where alpha amylase is, there will be lots of cellular material, so DNA evidence. So this helps you to localise an area, and then using a yellow, because obviously yellow and black are very good contrast, circle the positive areas. Using your note taking, and you may want to draw a diagram of your balaclava, you then might want to use a blue crayon and document where the positive stains and reactions you have obtained. Once this is done, you can then use swabs and target this area and swab that cellular material, that DNA evidence, and then send it off for DNA profiling. And that might maybe help you to identify whose that saliva is. Thank you for listening to the tutorials today. If you've got any questions about any technical detail or you'd like to speak to us, please contact the module leader, myself, Paul Langton, either on email or Teams, or just pop in, knock on the door and come and see me.